Thank you for joining the Once Changing the World, which is India's first future tech meets sustainability podcast. And today, I'm delighted and honored to have with me Mr. Akshay Maheshwari, who is a bioengineering graduate student working with Drew Endy's lab at Stanford University. He's also the co-founder of b.next Bio. And today, we're going to be talking about cell engineering. So, Akshay, I really appreciate you taking time being part of the podcast. Why don't we start with a small, brief introduction background?
Yeah, thanks, Eddie. Thanks so much. So Drew and I started working together first back in high school. I got really into programming computers, science, and one day I got invited to work in Drew's lab, and I realized we could program biology. And that's pretty incredible. Like, could we grow trees that had chips on them? Could we engineer the cells within us? It sort of opened my eyes in a different way. And so went to UCSD to do bioengineering, got really interested in how we could maybe apply that globally. How could we engineer world health? How could we scale synthetic biology globally? And so ended up coming to Stanford about 9 years ago now to do an MD PhD, where the idea was, how could we systematically approach biology and medicine in a different way than has been done so far, which is typically ad hoc based on our lack of understanding of biology.
Can you explain what synthetic biology is?
Yeah, synthetic biology is really a thought towards engineering biology. Before synthetic biology, we had really a different approach to genetic engineering, which was to understand biology. Synthetic biology is about building biology to understand it. And also to apply it for engineering ideas. So the field really took off in the early 2000s when we started engineering organisms to do simple things, whether it's blinking lights or simple oscillators. And since then has taken off to this idea of programming biology in the way that you might program computers.
You've been working with Drew Endy, who's considered as the father of synthetic biology. So how's it been working for Drew, and what are the cutting edge of innovations coming out of the lab?
Yeah, Drew's really a maverick, and that's kind of how the field got started, really. So Drew is wonderful in that he brings together people and creates contexts in which sort of new ideas and innovation can thrive. So when I started working with Drew in grad school first, it was really, what do you feel like working on? That was sort of the question posed. And there was this underlying idea of, what if we started thinking about biology and bioengineering from a bottom-up approach? Sort of since the start of synthetic biology, there's been this idea of, what would an operating system for biology look like? And we were starting to think of a new take on that idea, which was building that from the bottom up, as opposed to taking existing organisms and modifying them. So working in Drew's lab means, all right, let's go ahead and play with that idea. So for example, for myself, I said, back in the late 2000s, like 2009, 2010, there was this beautiful work around sort of a language for synthetic biology open language. People were designing these genetic circuits that could do incredible things, but it was really hard to implement them. It would take years to really get functions working. So when I went to Drew, I was saying, okay, what would it look like to really lift the lid on that? Look under the hood. What would a theoretical language for synthetic biology look like, and how would that operate? And so to directly answer your question, right, I spent a year and a half, 2 years trying different things. I built out a generative modeling system to try to create and dream cells from scratch. That didn't seem to be the right timing. So I thought about applying information theory and sort of lifting the structure of electrical engineering and bringing it over a little bit. That didn't seem to fit either. And we can dive into a little bit more, but what ended up happening was, by bouncing back and forth with Drew, saying, what about this idea? What about this idea? We came to the idea of physics and first principles physics as a way of understanding how all the molecules in a cell work, and thinking about applying that for engineering. So really Drew and Drew's lab is an amalgamation of these different creative ideas that start from scratch and then connect together to emerge into, for example, right now, a group of people who I'm really happy to be working with, who are thinking about building cells from scratch.
We have like a basic understanding, when we are creating an application in a software, we have a certain language you code with, you create an application, we get that. Designing life from scratch, how does one go about doing something like that? Can you elaborate on that?
There's two aspects to this. One is engineering biology, and the other is designing life. I think when a lot of people, practitioners in synthetic biology, they would say they're engineering biological systems to do useful things. So, for example, what is something that you would want to be real in the world? Maybe you want to be able to convert waste into food. Maybe you want to convert sunlight into energy. Biology is incredible at these things, which is why it's so powerful. And so we have a history of understanding how biology and cells can do incredible things. And so far, we've been leaning on that understanding to think about what new designs would be. So people have been, designing life or engineering biology for millennia. When we engineer corn to provide us with more nutrition, right? Crop science is sort of the oldest version of that. And so really what we're doing is taking that innovation 1, 2, 3 steps further and saying, okay, in the first iteration of synthetic biology, it's okay, how can we engineer bacteria to maybe detect different metabolites in our colon, and detect cancer? We're now thinking about this one step further and saying, okay, what are all the constituents needed for an organism to function and do useful things? So, for example, it might need to be able to make RNA. It might need to make protein. It needs to be able to have metabolism, to have energy. All these different functions all fit together. And so just like how you might design a car or a bridge with all these different materials that fit together to create an amazing structure, we're sort of thinking about cells in the same way now.
So when you say you're designing or engineering cells, what are the ingredients that it's needed to design a cell? How do you go about doing something like that?
So there's this idea of like abstraction layers in synthetic biology, which is, on one hand, you have atoms and you have quantum effects at the smallest part of a cell. At the highest level, you have essentially modules. So what are the ingredients of a cell at the module layer? A cell needs to be able to make proteins. It needs to be able to have a membrane to protect it from the environment. It needs to be able to transport in nutrients to feed itself. And if you wanted to do something interesting, it needs some sort of a program, for example, to, let's say, put a receptor that can detect foreign pathogens, for example. That's one level. Now, how does that compile down to more details? One example is, in order to make proteins, you need this molecule called a ribosome, which is essentially one of the most incredible protein RNA complexes in cells that can take information and turn it into proteins. You need tRNA, which are other molecules that help the ribosome do that. And it turns out, if you go on and on and on about this, you have 108 different molecules, for example, that are needed to go from DNA into protein. And if you keep going further, people have figured out there are 473 genes that make different proteins and aspects that are needed for a minimal cell. And that's work out of the Craig Venter Institute. So that's really how we're thinking about the ingredients of a cell. And then the amazing thing about biology is that it needs to fit together in the right way and have the right amounts. You can't add a kilogram of salt and an ounce of pepper. You need to add the right amounts. You need to add them at the right time. And they need to all be mixed together in the right way.
What can we do or build by gaining the ability to design cells from scratch?
Yeah, so there's a few things. I think the most exciting has to do with intrinsic control. So what do I mean by intrinsic control? When we engineer cells, typically, we have to deal with a system that's evolved over time to evolve more. And so when we engineer a system to, let's say, again, like detect cancer, it'll likely mutate away and you'll lose that program. One of the coolest things from building cells from scratch is you can actually change that basic code in a cell, for example, to not make it evolve. And so what does that actually mean in terms of applications? I want to be able to use cells to create smart RNA vaccines for emergent infection response. We had the COVID pandemic. It took us a year to really build and deploy those vaccines, even though we had the information perhaps to start developing them within a few months. What would distributed manufacturing of these vaccines or any therapeutics look like instead of having a centralized factory in which we then distribute the vaccine? One thing that synthetic cells can do are essentially act like a smarter version of that RNA vaccine by having all other components within it too. And you can imagine sticking a synthetic cell in your nose that can detect new pathogens that come into the environment, new strains, and can release, sort of protect you in response. You can also start thinking about live therapeutics. So it's not just detecting pathogens, but it's detecting autoimmune disorders, or it's detecting cancerous changes in your body. And sort of a third is bespoke production of materials. So at least in the US, there's a big push for example, manufacturing. How do you create new materials? How do you create new semiconductors? How do you create electronics? One power of biology is that you have proteins that are really good at making new materials. And so you can imagine creating materials that are stronger, more flexible, more oriented towards sort of like flexible materials, and really have tunable properties. So those are a few examples.
Everything's kind of made out of atoms, it's basic core, where's the magic transition? Where do these atoms turns into biological systems?
Yeah. Now, where the magic happens is at, I'd say, two levels. One is when atoms come together and bond to become proteins or RNAs that encode information and can do certain functions. So, for example, like a polymerase is a sort of protein that might be able to take DNA and turn it into RNA, which is two different types of information encoding. There's a second level of magic that happens when you group all of these together. So now you have groups of atoms that make molecules, and then you have groups of molecules that can make cell-like systems. And so now you have this, imagine this just crowded, packed world. It's like a gumball machine where everything is just moving around. It's super crowded. You're saying hi, they're saying hi, and they all work together to create this beautiful emergent function. You have somehow these tRNA moving through the crowd. They're finding the ribosome. They're building a new protein. And now all of a sudden, this new protein is maybe responsible for some of the conversion of light into growth and material. And so that's really the magic. It's when these emergent properties come from groups of molecules working together. And then one level higher than that is then all of that is under the hood of a cell, which can then propagate itself and do even more cool work.
What's the step? I'm sure there's too many complicated steps. How do you go about, like, is there a way to engineer life in itself?
What is a human, right? What would it be like to engineer a human? We have trillions of cells that build us up. It turns out half of those, or even more than half of those, are bacteria. There's our gut bacteria and bacteria all over our skin surface that make us up. And that's true for the world around us. If you have that wall behind you, there's going to be bacteria on that. This plant behind me, there's bacteria in that. There's also plant cells, maybe a little bit of fungi if I'm not taking care of it properly. And so when I say cells, it's because all of those together create organs, they create organisms. And so once we can start thinking about how to engineer cells, we talked about atoms, molecules, cells. The next level above that is groups of cells. How do, so our microbiome has cells that are all these different bacterial organisms. And maybe one interesting to note here is that the simplest organisms are just one cell. So you have bacteria that are just a cell, but they're also an organism, and they're all working together to do all the work in our gut. Those are groups of cells. And then there's also, like we said, humans or small organisms, where if you can start engineering the cells within them, you can start thinking about engineering those organisms. And yeah, what would that look like? Well, sort of, it's a green field. It's incredible, right? What would it look like to be able to start engineering even better organ systems? We have kidney transplants, we have liver transplants. What would it look like to engineer livers and kidneys that were one step above our existing organs, for example? And that's true for all groups of cells.
Lovely. How cool is that? So, I mean, your research is opening up a paradigm of things to be done, which could be just completely incredible for the future of mankind. You did, speak about the applications of, program cells. I mean, but could you kind of elaborate on, what could be the application specifically in the healthcare and medicine space?
Yeah, absolutely. And I mean, that's one of the big reasons I got into this. When we think about our approach to medicine right now, it's obviously been incredible. We've developed new drugs for cardiovascular disease, for metabolic diseases like diabetes, to everything from cancers and autoimmune diseases. How we come up with those approaches is scientific inquiry. We say, what if we took this new drug and tested it with thousands of people and see what it does? Or we invent cutting edge new therapies. For example, the newest therapy in cancers is cell therapy. It's something called CAR T therapy. It's a cell therapy where you can actually engineer cells to fight your cancer, essentially. But how does that actually come about? It's years, decades of engineering research. It's years of science to discover new pathways in humans where we say, oh, okay, if there's glucose here and there's a response to glucose here, what if we turn this one off? What if we turn this one off? How can we use that to improve the lifespan and health span of humans? What would a different systematic approach look like? And that's where sort of building from the bottom up comes into play. When we build a bridge, we used to look at a fallen log and say, okay, how can we cut pieces into this log to make a bridge? And then we had computer-aided design, where we thought about, okay, what would it look like to actually build this from scratch? So when it comes to health, I really foresee an evolution of our ability to build biology where we can systematically start thinking about what are the different components that build up humans that we can modify and manipulate and engineer to be resistant to different diseases. And so I'll give you a couple of examples. Cell therapies exist. They're incredible. They can detect everything from cancers to recently infections. But we're using existing human cells, for example, to do that. And so what that means is you can often have immunogenic responses. You can have rejection. I can't take my cells and give them to you to use because there's not going to be compatibility. Now, if you build cells from scratch, you might be able to solve some of that. You can create cells that aren't immunogenic. They're maybe universal cells. They're off-the-shelf therapy. What would it look like for you to walk into your local pharmacy and pull down, cell therapy from the shelf to deal with your, newest infection or your newest cancer? That's simply a different paradigm. but that's still within the engineering cell. Even more broadly, it's by building cells from scratch, we understand all the ingredients and pathways and the ways in which things can go wrong. That'll hold as we build more complex cell systems, as we build more complex organisms. And that'll not only give a direct engineering approach for putting new things in the human body, but also a deeper understanding of the pathways that make us up and the ways in which we can change it.
You had recently written a paper on engineering cell. Would you like to talk a little bit about that?
Yeah, so we recently published a paper on engineering tRNA abundances for synthetic cells and thinking about engineering biology. And that was really our connection of the theoretical world of physics and really deep in that stack. I sort of said you have modules, and that goes all the way down to atoms. What we proposed and what I've sort of worked on for the last 7-8 years with my colleagues is creating that physics engine for how life works. So it's a concept called colloidal physics. You have all these molecules, hundreds and thousands of molecules moving around by physics and then interacting, having reactions. So we built a model of that before this paper. And the idea was then, how can we use that paper to think about, and that concept and theoretical work, to think about engineering cells? And so what we did was we saw that recipe for a simple synthetic system. There are 108 different molecules in that. And what we said was, let's see what changing the amount of tRNA, which is a essential molecule for making proteins, what would it be like if we engineered that to really make a better synthetic system? So if I make an analogy to that, we were cooking a simple dish, and it was really taking the essential ingredients, again, salt, pepper, cumin, and saying how much of each, what is the perfect amount to make the perfect dish, and what is a perfect dish? So we created a CAD tool integrating some evolutionary algorithms that wrapped around our modeling to say, how do we make a completely different synthetic cell, for example, that doesn't evolve and does what we want, and make proteins really fast, or make it really slow? And what are the recipes for that? And can we design that directly? And so that's what we did. And then we actually implemented that system in real life, and it worked, which is incredible, because typically with these processes, it's months or years of iterating to do optimization and get it working. But when you build systems from scratch, they often just work. And so I'm really excited about this paper, because it's sort of that first example of really intentional engineering of these systems with computer-aided design and wetware. And sort of I'm excited to see how that continues to scale to, we did 21 of those 108 molecules, how that scales to more molecules and entire cellular systems.
In the beginning of the conversation, you mentioned about engineering cells and making trees create something else. So what are the crazy innovations which you think can come out of engineering cells?
Yeah, so that's I'll start by saying the most exciting ones, I don't know, because and I think that's what's most exciting to me. We're operating in the 1980s for computers, where you're asking me what's the coolest thing I can imagine, and I can't yet imagine the iPhone. I can't imagine the internet. I can't imagine distributed computing and machine learning. I don't know what that looks like yet, and that's what I'm so excited about. So what does the future look like in terms of crazy applications? I think we'll find out when 1 billion people are programming with biology. Some of the things that are in my head, which I don't think are the coolest, are things like distributed computing. What would it look like for the trillions of cells that are all over the world to be actively sensing and computing their environment and providing us with information? What would it look like to build organisms from scratch? We talked a little bit about this. Could we start changing sort of the basic functions that underlie us? What would it look like to have distributed manufacturing, food access, clean water access, medicine access is incredibly inequitable currently. There's so many regions that have a lack of basic resources. What would it look like if we could, right now you can make medicines and foods with yeast. If we can start building cells from scratch, what would it look like to be able to convert waste into food and sort of enable small communities to be able to use biology for their own problems without having to, talk to some researcher at Stanford? What would it look like for people to solve their own problems? I think those are like some of the most exciting for me. But even more exciting, I'm so excited to think about what they would create for themselves and for others, and sort of have that networked community that can create something incredible.
Lovely. How cool is that? I'm waiting for that awesome future to get into that awesome future. You mentioned about distributed computer. When you say distributed computing, are you hinting at biocomputing? Can you elaborate on that?
So biocomputing, it is what I was saying. It's powerful in two ways. One, in that a flask of cells can do an incredible amount of computation. If you have a billion, a trillion, even if you consider them as bits, you can start doing, in theory, an incredible amount of computation just in a flask. You can, right now, one way in which that's sort of implemented in the world right now is DNA memory. So, for example, capturing the entire Library of Congress in a little test tube of DNA. But one or two or three steps further than that is a flask that can do computation, that can do machine learning algorithms and propagation within a flask. That's cool in existing paradigms. One paradigm further than that is distributed biocomputing. It's activating the biology that already exists in the world around us to respond in our environment and do in situ computation. And that's going to be fundamentally different than essentially silicon or semiconductor-based computing, where it's very locally manufactured, locally implemented, or globally distributed, but it's not innately distributed. It's something really powerful that the world yet hasn't acted on, which is natively distributed computing and empowerment.
Are there any educational or training programs you recommend for people interested in pursuing a career in biotech, synthetic biology field?
It's a great question. And maybe this is where I'll again mention Drew Endy, who is right now running a course that's intended for not just university students, but a much broader community, and sort of building in those basic principles of synthetic biology. There aren't yet good global online courses, at least that I'm familiar with, for synthetic biology. So really, it's a matter of getting those roots with biotechnology. There's a competitive program called iGEM, which is all about how can we use synthetic biology for different applications, and it's sort of a competition that comes together every year. I would definitely recommend looking into sort of iGEM and some of the training resources that they have, and I believe it's online now. But some of the coursework that Drew teaches cells, it's a really good place to start.
Can you leave us with what the future is going to look like? Because there's so many cool things happening, and somehow all of these technologies are kind of converging. You can't say that it's just one tech stack. Metaverse is converging with Web3, it's converging with artificial intelligence, and so on and so forth. And same thing is happening with biotech stack. Could you paint a picture of how the world is going to look like in the next 10 years? Maybe the good part and as well as the scary part.
Like you said, right? I mean, huge convergence of technologies within machine learning, certainly, within VR, AR, within distributed currencies, with crypto, and within synthetic biology. There's the world in 10 years is headed towards really distribution, right? It's empowering distributed communities, whether it's through currency, biotechnology, machine learning, and computing. And so the world in 10 years, it's going to be using all of these technologies internally, and that's it's really incredible to imagine. We're already seeing so many innovations with machine learning, but as that starts to really touch biotechnology a little bit more, it starts to touch distributed finance a little bit more. I really see a world in which the individual and the community is much more empowered with sort of the means of productions and the means to contribute, and the means to empower themselves. Is what the really optimistic the future that I wish to see, where we're able to use these problems to sort of deal with inequity in a really powerful way, whether it's through enabling basic amounts of income, whether it's able to provide basic amounts of food, medicines, clean water globally. And then I think there's also maybe the side of it of just way more powerful technology, whether it's it's interesting because we talk a lot about self-driving cars, but I don't think that's not the biggest use, right? It's only helping a certain amount of people, but really, how does that then translate to transportation in rural communities? How does that enable better biotech where we can engineer these cells to be able to create new medicines? And so that's the powerful future I see in 10 years. I think a scary aspect of it is the complement to what I'm saying is if it becomes siloed, if, the only use for machine learning ends up being self-driving cars, or if the only use for biotechnology is creating the next expensive drug that only helps a certain amount of people. And so I think it's going to be a huge sort of tension, and I'm excited and optimistic for the future where we can create a culture of sort of sharing and empowering each other.
Akshay, really appreciate you taking time and being part of the podcast. We're living at a fantastic point of time, and I think we humans have the choice or the opportunity to, take the right steps because there's so much great things happening. We so far we've been living in a very centralized world, top-down, hierarchical, capitalism rules everything. We are opening the conversations of what a world could look like if it's decentralized, it's distributed, and everybody is empowered. Sounds fantastic. And I personally would want to see that world. Not so very sure about it because the world unfortunately is run by just a couple of percentage of the population. So, but I really hope that the world becomes decentralized. Wish you and your team the very best. And to my listeners, if you like what you see and hear, please press the subscribe button. Until next time, see you guys. Bye bye. Thank you, Akshay. Really appreciate this.
Thank you so much, Eddie.